Hi, my name is Nikhil Shah. I was working with Yifei Shu on applications of pulse electric fields for opening the blood-brain barrier. So, a little overview on our project. So as I mentioned, we are using pulse electric fields for opening the blood-brain barrier, which is a natural defense mechanism of the brain and the body to prevent pathogens from entering the brain. Since it's a highly sensitive area. And so there's this layer of endothelial cells that surround the brain, which we need to bypass in order for to have some practical applications in targeted drug delivery in more of a non-invasive or less invasive manner. So this kind of starts as a alternative to more invasive and dangerous methods such as Mire reservoir and things of that nature. So essentially what we're doing is we are using essentially less invasive ablation method to open and create small holes in the cell membranes, allowing some drug or some nanoparticle to pass through, and that's what we're measuring here. So a little bit about our experimental parameters. So we are applying pulse electric fields, and we are applying them at a ratio of 400 pulses at 20 hertz, 100 nanosecond pulse width, and 1,000 volts. And this is because we've previously shown that these PEFs never cause cell death over 24 hours, making them very viable for using the microscopic imaging, and they are able to be applied with the fluorescent tags, so we can also use the fluorescent microscopy to image the cells after a certain period of time. And we're also monitoring these cells at different intervals. And so as I've shown here, we're measuring at 0 minutes past the pulse electric field application, 0.5, 2, 4, 8, 24, and 48, and up to 72-hour time points after the pulse electric field treatment has been applied to the cells. And we're also using control wells as well. So these are going to be, about half of the cell array that we're testing, and these won't have any pulse electric fields applied to them, but they will be imaged and using the microscope and also the fluorescent microscopy. Moving on to our experimental setup. So on our side, our experimental setup was primarily mechanical and electrical things that needed to be set up. And so we were using a variety of different components. So as you may know before, we're just using a standard M6 breadboard, and we're attaching everything to this. So we are using 4Labs MLS sliding stage for our XY sliding stage, which you can control using the Kinesis software. And it is being moved in, it's a custom movement in single steps, 19 millimeters, which is the diameter of the wells, so we can move it in sequential amounts so we are centered each time. There's a variety of 3D molded, 3D printed components as well, which I designed, and I'll talk about a little more later. And we're using a generic stepper motor, which has a travel length of 24 centimeters. A 24-volt AC power supply, some control drivers, and a microstep driver as well. And we're using the Fuyu application, which is a kind of like a software application, a guided user interface application for the linear stepper motor, which we are using for controlling the depth of the electrodes in the cell wells. And we calibrated that to about 7.2 centimeters from the limited switch. And finally, we're going to be using the NanoKnife IRE electrodes, 2 of them, for the cell ablation, which we are applying the pulse electric fields. So here's some images about our setup. Moving on to our conclusions. So just a summary of our findings. The most up-to-date data, there is no significant difference in the transport of any specific type of nanoparticle when no pulse electric fields were used, regardless of the time point. However, pulse electric field pretreatment of the monolayer of cells significantly increased the transport of these nanoparticles at different time periods, an increasing amount over time, up to about 8 hours, as I mentioned before, 8 hours after pulse electric fields were applied. And this is just kind of a diagram of what our setup look like for the cell wells. So we have this transwell, which is this kind of membrane permeable layer here. The cells are applied here, and we are applying the nanoparticles as well. And we have an apical and a basolateral layer of cell culture in which these fluorescently tagged nanoproteins over here, or nanoparticles over here, are passing through this membrane. And we can recover this and measure the concentration of this in this small sample of the basolateral cell culture down here. And as I said before, that we're using 400 pulses, pulse electric fields, and we are imaging up to over 24 hours, so up to 72 hours actually. Past pulse electric fields have been applied, and we're getting good results. That's the main thing. We're getting extremely good results. We're seeing a positive correlation in pulse electric field between pulse electric field application and nanoparticle transport over certain periods of time, and it's not causing extended cell death, immediately after treatment, which is good because otherwise we wouldn't be able to measure anything. And finally, a little bit of notes about how our data was collected. We're measuring using a one-way ANOVA. This is called, I guess, a Boniferroni test and a Turkish test. So we can measure the same level of significance of each individual nanoparticle in terms of the amount of nanoparticle concentration passing through these cell membranes over time. Okay. And that is all I have for you today. This is just a summary of my research for this quarter. We will be continuing on next quarter, but I think you guys would be happy to know that we are coming close to finishing up our paper on this, and it seems like this experiment will come to a close pretty soon. So have a nice break. Thank you.